going to talk about the project that the title you can see, but before I start my talk, I want to give an appreciation for international students that I think have been suffering from the interaction of immigration policies and the pandemic together has been hard on many international students. So I am really lucky that 2 international students have joined my lab. Here is a picture of us with his picture of Bin Wang. So Bin Wang has been working with me on networks undergoing synaptic plasticity, and his paper came out in PRL over the summer. His new project will be presented at SFN next week. And Palka, whose work I will talk about today, and she's here with us. Her project, again, you'll hear about today, and she's also presenting at SFN. All right, so the outline for my talk, we're going to talk about fruit fly olfaction, and the talk will be in 3 parts. In the beginning, I will tell about some preprocessing that happens in the periphery of the olfactory system of flies. There are these receptor neurons that are intriguingly coupled, and with our collaborator Qing Su and Shanse Wu, we built a model for this preprocessing. After I will tell you about this preprocessing, we will look into what are the implications of the preprocessing of olfactory signals that happens in the periphery. So it's very well known that from the antenna that are shown here, olfactory signals are relayed to the antennal lobe, and then further relayed to 2 centers in the central brain, 2 regions in the central brain of flies, the mushroom body and the lateral horn. And we will show that the preprocessing that happens in the periphery has some implications that we can test in what goes on downstream. Okay, so this is a picture of an antenna of a fruit fly, and you can see here a bunch of these hairs that are called sensilla. In each sensillum, there are typically 2 receptors, 2 olfactory receptor neurons, and I'm going to use the abbreviation ORN. Each ORN is selective for a particular odorant profile, and we're going to assume that each ORN is selective to 1 odorant. Intriguingly, the ORN types that are coupled in the same sensillum are preserved. So, for example, in sensillum AB4, you always find ORN 7A and ORN 56A. In ORN AB8, you find, in sensillum AB8, you find ORN 43B and 9A. So it seems like evolution is working very hard to preserve this organization, to put specific ORN types together, presumably for some kind of olfactory processing. And this was a question for a long time, especially because despite this conserved organization, there is no synaptic connections or gap junction connections between these ORNs. So evolution is working hard to put them together, but then it seems like, or for a long time, it seemed like there was no effect that they were having on each other. So our colleague Qing Su, in her work in 2012, showed that despite the fact that these neurons don't interact through synapses, they do have ephaptic or electrical interactions, and they inhibit each other through these electrical interactions. So this is a cartoon of a sensillum, and when you present ORN A is the large one, you can see here the large spikes of ORN A and the smaller spikes of the smaller ORN B. When you present the animal with the odorant that ORNA is selected for, then ORNA starts firing at a high frequency. But then when you present on top of that the odorant that ORNB is selected for, ORNA shuts down because of this interaction, and ORNB starts firing. Importantly, it was also shown in this paper that these interactions are important for behavior. So I'm not going to go through the details of this experiment, but essentially what was done is to block the output of one of the neurons, the A neuron here, but leaving intact the interactions that happen at the dendrites, at the level of the dendrites. And what was shown is that the signal, when neuron A is stimulated, it inhibits neuron C, and that conveys enough of a signal to influence the animal's behavior, even if neuron A does not spike and does not by itself transmit information downstream. So I'm going to skip the details of these experiments. If you're interested, this has been published 10 years ago. Another important clue is that Ephaptic interactions are known to depend on the ORN size. We can think of this as kind of a nonlinear inductor. So the strength of the interaction depends on the size of the two ORNs. And not only are the not only is the identity of the ORNs that are coupled preserved, also the relative size. So Chi-Ying's lab in 2019 showed that if you look at multiple copies of AB3 sensillum, AB4 sensillum, and so on, the soma volume, the inner dendrite volume, and other morphological quantities of these neurons are also preserved. So this suggests further that the interaction, which neurons interact, is preserved, and also the nature of that interaction is highly preserved across animals. Okay, so this is what we want to understand. Why is there such a stereotyped organization? Or in other words, what is all this peripheral preprocessing good for? And we're going to build a model for that. One reason, before I get to the model, sorry, another clue for why this peripheral preprocessing is important came from a paper that Chi-Ying published earlier this year. We characterized in this paper the behaviors that are mediated by the large spike ORNs, the larger of the ORNs that are paired, and the small spike ORNs. And very consistently, you see that large ORNs are selective for odors that have positive value for the animal, and small ORNs are selective for negative value odors. So, for example, in the AB4 sensillum in female flies, the A neuron promotes egg laying in females, and the paired neuron B suppresses egg laying in females, and a similar thing happens with courtship behavior in male flies. So a hypothesis that comes out of this work is that Ephaptic coupling between ORNs is important for extracting odor valence. And this is the hypothesis that we're going to try to test using model and additional data analysis. Okay, so we need a model, and our model, one requirement for our model is that it will include neuronal responses to turbulent or transient stimuli. In the results that I've shown you thus far, stimuli were presented for a duration of about half a second or more, but these animals are known to fly through turbulent odor plumes. So the concentration fluctuations of odorants that they experience look much more like what's shown here, not these blocks of odor presentation for 2 seconds. And for technical reasons, it's challenging to present these kinds of odors to animals. So we're going to do that in the model. Okay, so we want a very simple model because there are about 50 types of sensilla, and we want the model to be tractable. So we thought we might start with a linear model for these transients, because linear models, the transient behavior of linear models was shown by many people to be interesting. You can get interesting amplification behaviors in this scenario. There are nice papers by Ken Miller and by Serjan Ostojic. But when we did some further reading into the biophysics of these ephaptically coupled neurons, we understood that we have to include some nonlinearity in these interactions. So the model is we have transient decay to baseline. We have coupling that we're going to replace the linear coupling with nonlinear coupling. And the stimulus, importantly, is time dependent. So these S's are going to be functions of time. So we replace the interaction, the linear interaction, with nonlinear interaction that has two important forms of the nonlinearity. This is what our equations look like. So we have, first of all, the effect of neuron B on neuron A is nonlinear. So there's this nonlinear exponent n. This mimics the threshold effect that neuron B has to be active beyond some amount for neuron A to feel the inhibition. And the second important nonlinearity that we have is that the inhibition that neuron A experiences depends on its own level of activity. And these forms of nonlinearity are supported by the experimental literature. We have two parameters in this model. Q is a very important parameter that I'm going to come back to later on again and again. Q describes the asymmetry of ephaptic coupling. So neuron A, the inhibition that neuron A experiences is weaker by a factor Q relative to the inhibition that neuron B feels by the activity of neuron A. And the second parameter is the degree of nonlinearity. The fact that this model is so simple, even though it's nonlinear, we were able to solve it analytically when the stimuli are pulses of odor concentration. So we imagine a scenario where there's a pulse of odor, odor A and odor B, and that sets the initial condition. That sets the initial condition of the firing rates of neurons A and B, and we solve the decay to baseline of these firing rates. So that was the first advantage of having a simple model, and the second advantage is that we were able to fit these parameters to electrophysiological data that we got from Qing, and I'm going to skip the results of this fitting. If there are any questions about the fitting or the parameter values that we got, I'm happy to answer that in the Q&A session. Okay, so we have these solutions. Now let's try to test our hypothesis with these solutions. So we started by thinking that ephaptic interactions might be important for processing the value or the valence of an odor. So we defined this quantity, alpha Q, which is the ratio of this weighted combination of the firing rates of neuron A and neuron B. The weight here is Q to the power 1 over n, which I'll come back to in a moment. So this is a ratio of the this weighted linear combination of the firing rates relative to the the same linear combination of the stimuli at time 0. And the source of this weight factor is that for this weight factor, our analytical solutions for the dynamics show that there is equal inhibition from neuron A to neuron B and vice versa. So if we think of neuron A as conveying a positive value signal and neuron B conveying a negative value signal, then these two neurons inhibit each other equally when we have this scaling factor based on our solutions to the model. So we interpret this quantity as the transient net valence of an odor, and we compare it to the net valence of the odor at the time of simulation. Okay, so we define this quantity, and we have the solution, so we can compute this quantity analytically for different parameters and different values of the stimulus. And what are we going to do with it? So at time 0, this, by definition, this quantity is equal to 1 because the stimuli set the initial condition for the firing rates. At time 0, these neurons, the firing rates decay to 0, so the numerator is 0. So this amplification quantity is equal to 0. And the idea is that maybe at some intermediate time, this quantity is greater than 1, and that could potentially lead to some triggering of a behavior downstream if the valence signal is amplified strongly enough. So we compared the transient decay of firing rates in this model when we don't have interactions. That's shown in black, and we have a straight line decay to 0. When we do include interactions, then this trajectory is curved because these neurons interact with each other. And when we plot this quantity, alpha q, we see that this quantity suggests that this valence signal is amplified when there is interaction and not amplified, decays monotonically to 1 when there are no interactions. Importantly, this amplification is stimulus specific. So if we look at the same strength of the interaction, but a different mixture of these two odors, then the degree of amplification is much weaker. So this circle here represents the fact that we're looking at stimuli that have the same overall amplitude, but this stimulus is amplified much less than the solid line. Okay, so we identified a mechanism for amplifying odor. The next thing we did is to look whether these interactions can aid in odor discrimination. To do that, we defined the response angle, which is time dependent because the firing rates are transiently changing. And we compared nearby initial conditions in brown, orange, and yellow. You can see that the initial conditions or the stimuli that are in brown and yellow, the trajectories don't separate before they collapse onto each other. But in orange, there is some degree of separation before the firing rates collapse to 0. We can plot the relative change in their angle as a function of time relative to the difference in angle at time 0. And for the orange initial condition, for the orange initial condition, these trajectories, this curve shows that these trajectories separate. Okay, so we understood something about how these ephaptic interactions can aid in odor discrimination. This is also stimulus specific. So the plots in the previous slide were shown for a particular value of this parameter Q, which is, I'm reminding you, the asymmetry between the effect of coupling from neuron A to B relative to B to A. So when Q is equal to 1, the system is completely symmetric, and the order that is most well discriminated is the mixture, the one-to-one mixture. But if we reduce Q to be less than 1, because ORN A is greater, is bigger than ORN B, then the stimuli that are best discriminated are in this part of the firing rate space, or the stimulus space. And this should remind you of this result by Qing's lab, showing that not only is the identity of neurons that are paired conserved, also the relative size. So it seems like the system is tuning this parameter Q such that particular odors can be discriminated. So we have some understanding based on this model, and we are now going to try to generate some predictions and test them in data. Okay, so our prediction is that downstream readouts should be tuned to take advantage of this transient amplification. And in particular, in this transient amplification, we had this weighted sum. So we want to look for a signature of downstream connections implementing this weighted sum. So to do that, we turn to the fly connectome dataset from Genelia Research Campus. So this is the hemibrain dataset, where a big chunk of this animal's brain was the wiring diagram was extracted. And we're going to focus on a particular part of this wiring diagram. We're going to look at the connectivity matrix involving the antennal lobe, which relays signals to the lateral horn. And within that big matrix, we're going to look at a particular part of it, which is shown here. So this rectangle represents connections where the antennal lobe neurons are presynaptic and the lateral horn neurons are postsynaptic. So because our model can tell us something about how the signal is prepared at the level of the antennal lobe, we look at feed-forward connections from the antennal lobe to the lateral horn. And within that class of connections, we look at a subset. So you see that this entire rectangle is connections from the antennal lobe to the lateral horn, but we only focus on this smaller rectangle. The reason for that is that the neurons, the projection neurons from the antennal lobe to the lateral horn here carry signal from a single glomerulus in the antennal lobe. So I can give you a very brief description of fly neuroanatomy. Each ORN has its private glomerulus in the antennal lobe. And from the antennal lobe, there are projection neurons to the lateral horn. So if we look at neurons that have that carry signal from a single glomerulus, we can think of them as carrying signal from a single ORN type, which is the quantity that we have in our model. We know something about the interaction between firing rates of individual ORN types. Why are we looking at the lateral horn? So the lateral horn is implicated in controlling innate behaviors, and there is conserved connectivity between the antennal lobe and the lateral horn. So since our prediction has to do with valence that the animal cares about innately, this is the natural target to look at. The conserved antennal lobe to lateral horn connectivity is in contrast to connectivity from the antennal lobe to the mushroom body, which was shown to be random by this beautiful paper by Sophie Caron in 2013. So I'm going to get back to the mushroom body at the end of the talk, but for now we're focusing on testing our prediction in the lateral horn. Okay, so what are we going to do? The first thing we did is to take this rectangle matrix and compute the PCA, perform principal components analysis on this rectangular matrix. And what principal components of this matrix mean, these are projection patterns from the antennal lobe to the lateral horn. And when we compared these principal components, the variance explained by these principal components to shuffles, we saw that the first 4 or 5 principal components explain significantly more of the variance than you would expect from random shuffles. The random shuffles here are not completely unconstrained. I don't have time to go into the details of how we did the shuffles, but this is, again, something I'm happy to answer questions about in the Q&A. Again, and this should be compared to the same analysis that was done in the mushroom body by Caron, and they showed that the principal components there can be entirely explained by the random shuffles, and that was based on this result. They argued that the connectivity from the antennal lobe to the mushroom body is random. Okay, so we identified some patterns of connectivity from the antennal lobe to the lateral horn that are non-random, and we want to look further into them and see whether we can find signatures of this readout that is predicted by our model of the processing in the periphery. Okay, and so in particular, we looked at the coefficients of each ORN type in the least random projection pattern. So the principal component 1 coefficient of each ORN, that is the weight with which that neuron's firing rate is being read out in the lateral horn. And we have a particular prediction for that, which is to compute net valence, this has to be that factor q to the power 1 minus n, 1 over n. Okay, so the first thing we did is to look at the statistics of principal component 1 coefficients for positive valence and negative valence neurons. And we fit a gamma distribution to these two distributions. For the positive valence neurons, we found a shape parameter for the gamma distribution, which is greater than 1. This means that the mode of this distribution, the most common value, is some positive value. The shape parameter fit to the negative valence neurons is less than 1. This means that the mode of this distribution is 0. So we already see a very significant asymmetry in how signals are being read out from positive valence relative to negative valence neurons. And we also compared the mean principal component 1 coefficient between positive and negative valence, and we subsampled the data to show that this result is significant. So still, this doesn't prove our point, right? Because not all the positive valence neurons are coupled with all the negative valence neurons. There are particular pairings that are conserved. And our model says something about how these particular pairings should be read out downstream. So this is what we're going to look at next. But at least at the level of the population between positive and negative valence neurons, this is what we would expect, that the system is tuning the readout to this direction of the stimulus space. Okay, so we want to look at more carefully at the angles, at the readout angles, and compare that to the morphometric asymmetry. So what we did is to, and the idea here is that by changing this asymmetry parameter Q, the system could tune the stimuli that it is most sensitive. So what we compare here in purple is the probability distribution from pairs of ORNs in our data. We're showing their readout angle. And we see that when we compare that to shuffles in pink, where we shuffle but conserve whether the neurons are positive or negative valence, we see that these distributions look different. In particular, there are missing angles around pi/6 and some excess angles at pi/3. But because we only have 26 pairs, this difference is not statistically significant. And in the paper that we're writing up, we have random matrix theory analysis estimating how much more data we would need for this test to be statistically significant. So we think that if someone if somebody extracts the wiring diagram for 1 or 2 more flies, we should have enough statistical power to show that the purple and the pink distributions are different, meaning that the readout angle is specifically tuned to the optimal stimuli in the periphery. But this is not the end of the story. We did another analysis that did reveal a coordination of what happens in the connectivity from the antennal lobe to the lateral horn and what happens in the periphery. So let me walk you through this analysis. We computed a compound morphometric ratio. So we have a bunch of size measurements of the ORNs in the periphery. We have there the volume and the surface area of the inner dendrite and the outer dendrite. So from these 4 quantities, we computed the linear combination, which is most strongly correlated with the readout angle. So because the Ephaptic coupling depends on the size of these neurons, we hypothesized that some compound morphometric ratio will be correlated with the readout angle. So here we fit the coefficients of this compound morphometric ratio to be maximally correlated. But then what we did is to shuffle the identity of neurons, preserving whether they are positive or negative valence, but breaking their pairing into sensilla. And when we do that, then the same correlation is significantly weakened. So the correlation that we found between the morphometric ratio and which is something that is this is a quantity that exists in the periphery, in the antenna of the fly. This is significantly correlated with the readout angle of the odors that these ORNs transmit signals of much deeper in the brain. So this angle lives in the connections between the antennal lobe and the lateral horn, which is a number of synapses downstream. Okay, so that's the end of the part on the lateral horn, and I'm going to move on to the mushroom body. Those of you who have been following the mushroom body literature might ask, didn't we solve the mushroom body already? So in this beautiful paper that I mentioned a couple of times already, Caron et al. Showed that connectivity from the antennal lobe to the mushroom body is random. And this has been investigated by a number of papers since 2013, and people report some degree of structure in these connections, but that structure, I hope we can agree, is very, very small, and by and large, we can say that the connectivity is random. 1 year later, Bakhtash Babadi and Chaim Sompolinsky published a beautiful theory paper showing that if you have an input layer with that represents stimuli that are high dimensional but not very high dimensional, let's say 50, that from that input layer, the stimulus is then expanded randomly, and the responses in the second layer are sparsified, then that kind of computational scheme they showed in this paper is good for learning associations between novel odors and the appropriate response. So if the animal is presented with a novel odor, using this random connectivity, which expands and sparsifies the signal, you can learn to classify new odors in a way that is better. So these two papers seem to go hand in hand, and in fact, in this paper, they had this cartoon where they showed inputs in the clustered inputs in the antennal lobe, and their interpretation in Babadi and Sompolinsky of these clustered inputs are odors that have maybe a little bit of noise. So if you have some odor percept, but maybe the concentrations change by a little bit, or the ratios of the different odorants change by a little bit, these are in the antennal lobe represented by points that belong to the same cluster. And then this matches nicely with the random connectivity, which then allows at the level of the mushroom body for better classification. A very big assumption in this model is that the inputs have to be clustered for this scheme to work. If the inputs at the level of the antennal lobe are not clustered, then we have a problem. And we indeed do have a problem, because some papers, both in flies and other insects, show that inputs to different odors at the antennal lobe are not clustered. So in these two papers, the conclusion that comes out of them is that responses to the same odor mixture at a different concentration can be much further apart in the when you look at antennal lobe responses than responses to different odors at the same concentration. So this is shown by this cartoon that the purple triangle, the big purple triangle, is further apart from the small purple triangle than the small purple triangle and the brown circle. But still, this is a very nice idea that the random connections can help in new odor classification, and experimental research of the mushroom body does show that this is important. This region is important for learning associations of new odors in flies. So we wondered whether we can use this pre-processing step by Ephaptic coupling, which was ignored in this model, to understand what is the role of random connections from the antennal lobe to the mushroom body. So let me present our idea first with a cartoon. Let's say we have a uniform stimulus distribution, which is shown here in 2 dimensions. We can think of the different interactions between ORNs in the periphery as sort of slices through this stimulus space. And so the Ephaptic interactions cracks this stimulus space, and they push points away from these cracks, such that maybe on one side of the crack we have positive valence or stimulus in one category, and on the other side we have stimuli in another category. And our question was whether these cracks are sufficient for this computational scheme of expand and sparsify to work. So I'm skipping some details here in the interest of time about how we assigned labels. So here the labels are assigned based on cluster identity. We assigned labels based on the primacy set, which is an idea that has been in existence in olfactory coding for a number of years. I'm not going to go into that, but I'm happy to answer questions about that in the Q&A. Okay, so we use the primacy set to assign labels in the case where the stimuli are not clustered, they are uniformly distributed. And we are then going to test whether these random connections are good for anything. Okay, so before I show you the results, we need to make a sanity check, or I need to prove to you that I'm not cheating. So we compared the distance between stimuli that have the same label or different labels when the inputs are clustered. This is how this would look like in 2 dimensions. And you can see here that this little bump in this distribution represents odors that belong to the same cluster. So all the odors that belong to the same cluster have the same label. So only the red distribution has this little bump at small values. When we look at initially a uniform distribution of stimuli, which is then transformed by Ephaptic coupling, the distributions of responses that have the same label or different labels are much, much closer. Okay, so this shows that we are clearly there is some effect for the Ephaptic coupling. They push stimuli away, but this is not a clustered distribution. Okay, so then we did some numerical simulations. In Babadi and Sompolinsky, they did analytical calculations, but because we don't have clustered inputs, then we don't know how to compute the classification performance analytically like they did. We computed the classification performance as a function of 2 parameters in the antennal lobe and 3 parameters in the mushroom body. So let me start with the antennal lobe. We have the amplitude of Ephaptic interactions, and this is parameter A, and parameter K is the range of Ephaptic interactions. The way to think about it is how far away does the stimulus have to be from these cracks to feel the interaction. And in the level of the antennal lobe, we found an optimal pairing of these 2 parameters that leads to a relatively modest improvement in the classification performance of odors. But when we then expand and sparsify the inputs, and the 3rd parameter that we use in the mushroom body is 0.2, then we get almost a factor of 100 improvement or so. We get 2 orders of magnitude improvement of classification in the mushroom body, only if stimuli first are preprocessed by the Ephaptic coupling. So we also tested the effect of the sparsity. So this point here is the point here. That's the optimal degree of sparsity that we found in our model that is not in that is consistent with the degree of sparsity of responses to odors in the mushroom body, and this shows the improvement relative to with all these comparisons. So we look at antennal lobe with no coupling, antennal lobe including coupling, and mushroom body with and without coupling. So our conclusion from this is that if stimuli are not clustered, then Ephaptic coupling can have enough of an effect, which is a little bit like a triage clustering, like a quick and dirty way of clustering initially unclustered inputs that makes the expand and sparsify computational scheme that happens from the antennal lobe to the mushroom body work much better. Okay, so I'm running out of time, so let me conclude. In the first part of the talk, we showed that ephaptic coupling in the sensory periphery can enhance the valence signal of odors, and the classification of nearby odors can be improved in a stimulus-specific way, as it depends on this parameter Q. So if the readout is correctly aligned, we get these benefits from ephaptic coupling. So, and then in the second part of the talk, I showed that we found signatures of this predicted readout pattern in connections from the antennal lobe to the lateral horn by finding a significant correlation between this quantity that exists in the periphery and this quantity that exists in the connections from the antennal lobe to the lateral horn. And so we think that we can say, based on this analysis, that the fly connectome implements a general purpose computation, which is to extract an odor valence for odor mixtures, not just for binary mixtures of odors. So for arbitrary mixtures of odors, this connectivity with the ephaptic coupling extracts an odor valence signal. And then in the last part of the talk, I turned to the mushroom body, and I showed a computational model suggesting that ephaptic interactions can perform a kind of triage clustering, which facilitates classifications of new odors by the expanded sparsified connectivity that happen between the antennal lobe and the mushroom body pathway. So before I take questions, I really want to thank my lab, but primarily Palka Puri, who has pushed this project forward, and I'm very happy to be presenting this work. And our collaborators, Qing Su and her student and now postdoc, Shanse Wu. I want to thank my other collaborators and funding, and if this kind of work is interesting to you, my lab is looking for students and postdocs to join, so please send me an email if you are interested. Thank you.